In this video, we'll talk about cardiac muscle, especially how it differs from skeletal muscle and the way that it's regulated. So if we take a look at just the myocardium, okay, the muscular wall of the heart, there are two types of cardiac muscle cells. The majority of the cells, 99% of the cells, are just the cardiac muscle cells that you looked at last semester when we looked at different muscle types in lab. Okay, so those are the contractile cells. And then there's another cell type, pacemaker cells, also known as autorhythmic cells. And these cells are very different, and we'll take a look at these two cell types separately. Okay, so first starting with cardiac muscle, which we're a little bit familiar with already. I'm going to jump over to picture. So here's a picture of cardiac muscle from our book, and you can see, hopefully you remember this from lab, that the cardiac muscle cell shape is a little bit different. The shape is described as bifurcated. In other words, you know, like you'd have one cell that splits into kind of like two branches. You know, that's what's meant by the bifurcation. So the bifurcated cells, it is striated muscle. You can see the striations, okay, that banding pattern within the cell. And where one cardiac muscle cell contacts a neighboring cardiac muscle cell, this area, this is known as an intercalated disc. So you can see the intercalated discs in this diagram. So that's just where one cardiac muscle cell is contacting a neighboring cell. And the significance of the intercalated disc is that that's where there are gap junctions. So again, this is a lot of AP1 recall here. A gap junction, here they're showing us a little depiction where this would be one cell, this would be the neighboring cell, and what they're trying to show us is there's basically like a protein that forms a continuous channel between two different cells. So that's the significance of the intercalated disc. In the intercalated disc, ions, as long as they're small enough to cross the gap junction, are going to be able to cross the gap junction. And so this, even though you have two separate cells, it creates like a functional connection between the two cells, which is very significant. So jumping back to the notes. Cardiac muscle cells are striated. Okay, we looked at that, bifurcated, or, you know, the branching structure kind of shaped like a Y. They attach to the neighboring cells in this specialized region called an intercalated disc. And the significance of that is that's where gap junctions are, which are tiny pores that connect the cytoplasm of neighboring cells. So as far as how cardiac muscle works, I would say that there are two main things that make it very different from what we talked about last semester as far as skeletal muscle. So before I show you those two things, this is how it is, you know, like what we'd expect. These cells maintain a resting membrane potential over time. Okay, so they only contract when they're stimulated to do so. So that, you know, we would expect. Pardon me. And when they're stimulated to do so, that just means that they were depolarized to threshold. But here is where there's a big difference. So once one cardiac muscle cell depolarizes, instead of that being like an individual event with a single cell that we talked about like in skeletal muscle, what will happen is, let's say if you had sodium come into the cell, those ions will cross the gap junction. And so what that's going to do is it's going to automatically depolarize connecting cells. And keep in mind, one cardiac muscle cell is attaching to two cells because of that Y shape or the bifurcation. And then those two cardiac muscle cells also split and they're connecting to four cardiac muscle cells. And so what's happening is there's an interconnected network within the myocardium that connects all of the cardiac muscle cells. So that as long as you can get one cardiac muscle cell to contract, that is going to sweep through the whole heart. And so that's very, very different from skeletal muscle. With skeletal muscle, remember, each individual muscle cell was controlled by a neuron. And so skeletal muscles worked in units called motor units. You might remember that. So you'd have to have neurons fire and stimulate the muscle cells that they're connected to, and it would only stimulate those individual cells. But because of these gap junctions and that interconnected nature of cardiac muscle, the heart is going to work as one unit. That's known as a functional syncytium. And so instead of having motor unit contraction like we talked about last semester, when the heart contracts, the whole heart contracts. So you're going to have an organ level contraction, not just, we're not going to have like just, one atrium contract and so on. So, different parts of the heart. That's not the way it's going to work. So that's very different. And here's the other very different thing between cardiac muscle and skeletal muscle. With skeletal muscle, when we stimulated skeletal muscle, remember it would contract and then it would relax, something called a muscle twitch. You might remember that. If we stimulated skeletal muscle frequently, then what could happen is if we stimulated it frequently enough, then the cell could contract and it could stay in a contracted state, right? That's known as tetanus, or even, partial tetanus. So that does not happen with cardiac muscle. So cardiac muscle behaves a little bit more like a neuron. You might remember an absolute refractory period in the neuron where if the neuron axon received another threshold depolarization while the voltage gated sodium channels were open, it couldn't respond. So in other words, there's a point in there, action potentials, you could not have an action potential fuse into one prolonged, event like you can with muscle. And so cardiac muscle works more like that, where there's an absolute refractory period of these cells. And just to take a look at a picture from our book, or a graph technically, what they're showing us here, the purple line is contraction. So this is what we were calling a muscle twitch last semester. Cardiac muscle contracts and then relaxes. And this blue area, the darker blue area, this absolute refractory period, would be a point in time when it's unable to respond to another stimulus to contract. So even if it was receiving frequent stimuli to contract, do you see how, where is this blue intersecting? I guess right around here. So it looks like by the time this cell, the cardiac muscle cell, could respond to another stimulus, do you see how it's well into its relaxation period? So you can't have tetanus in cardiac muscle. Thank goodness, right? Because with cardiac muscle, you want it to contract and relax because it's pumping blood. We want it to contract, eject the blood out of the heart, and then relax, fill up with more blood, contract, eject the blood. We don't want to, say, like in a fight or flight response, have the heart stimulated so much that, say, it contracts and it stays contracted. That wouldn't move any blood. So I hope this makes sense. So taking a look at the notes, those are the two main differences, I would say, between cardiac muscle and skeletal muscle, that, you can't see tetanic contractions because basically the absolute refractory period is going to last well into the relaxation of the muscle contraction, and that the whole heart is going to contract as an organ. So remember, just go back up here for a second. We said that most of the heart are cardiac muscle cells, these contractile cells. That's what we just talked about. Okay, the contractile muscle structure, the contractile cell function. So now I'm going to move on to these pacemaker cells, also known as the autorhythmic cells. So this, remember, these cells only make up 1% of the heart. So these cells are woven throughout the heart. They're highly specialized. Okay, the pacemaker or autorhythmic cells. They're similar to neurons in that they can generate action potentials. Okay, but they're different from neurons in that they can't maintain a resting membrane potential over time. These cells, the pacemaker cells, have slowly calcium channels, and so that means that calcium is going to continually enter the cell. And when a positively charged ion enters the cell, that creates a depolarization, right? The inside of the cell becomes more positive. And so because it's continually depolarizing, what will happen is eventually that cell is going to reach that threshold depolarization, and it's going to elicit an action potential. So because of these leak channels, these cells depolarize in a predictable and a rhythmic way. So because they're continually drifting towards threshold, what they do is they regularly fire action potentials, and when they do, that's going to trigger cardiac muscle cells to contract, and all of those cells are interconnected, and so that stimulus to contract is going to sweep throughout the heart. So that's why they're called autorhythmic cells. They're going to generate a pace of depolarization of the heart, and because of that, our heart has the innate ability to beat. Okay, we don't need the nervous system to tell it when to beat. You know, it's kind of crazy, but we'll talk about the nervous system too. So these autorhythmic cells, they're not just here and there. They form an interconnected network. They're all connected together. And so what I have in the sequence of excitation is basically the map of these autorhythmic cells. So I'm going to switch to a picture and show you where these are located. Okay, so in our book, what they do is they give us a picture of the heart with these areas in yellow. And the yellow areas are the autorhythmic cells. So there's a clump of these cells in the right atrium. That's called the sinoatrial node, or the SA node. And from there are fibers that branch out. This is a little just more diffuse into both atria. People sometimes call that the atrial syncytium or the internodal pathway. But the SA node, you know, will sweep throughout these fibers that go through the atria and interconnect with what's called the AV node, also in the right atrium. So in the right atrium, there are two clumps of these autorhythmic cells, the SA node and the AV node. AV node, you can see it's right on the boundary between the atria and the ventricles. The AV node is attached to a bundle of autorhythmic cells that cross through where the valves are located. So remember, the valves are located in that single plane, which is a tough, kind of fibrous plane. So this is where the message for the heart to contract is going to get through into the ventricles. It's going to enter into the interventricular septum. Within the interventricular septum, you can see it branches into the bundle branches, and then when the bundle branches hit the apex of the heart, big fibers called Purkinje fibers come up and innervate the walls of the ventricle. And so this is the path of the autorhythmic cells. This is what's known as the cardiac conduction system, or the intrinsic conduction system. So the intrinsic conduction system or cardiac conduction system is just this path of this map of autorhythmic cells. So to go back to the notes, you can see the SA node is at the top. That connects to the atrial syncytium, those fibers that branch throughout the atria, connects to the AV node. The AV bundle is the place that shuttles this message down into the interventricular septum. AV bundle branches are found in the interventricular septum, and then the Purkinje fibers go up the ventricular walls. So I made a note here about the intrinsic rate of depolarization, and you can see it's different in different areas. So in other words, people have, you could dissect out the SA node and put it in a petri dish. And if we could dissect out the AV node and put that in a separate petri dish, and dissect out the Purkinje fibers and put that in a petri dish, what we see is that these autorhythmic cells, they all work the same way, but they have a different rate of depolarization. So that the SA node depolarizes the quickest. Okay, so the SA node was right at the top of the right atrium. That depolarizes 80 to 100 times per minute, compared to the AV node that's slower. And you can see that the Purkinje fibers even slower. Now keep in mind, because these autorhythmic cells are all woven together, and that the cardiac muscle cells are also, interwoven into this functional syncytium, whichever area is fastest at sending out a depolarization, that will set the pace of the heart contracting. And because the SA node is the quickest, it will set the pace. And that is the pacemaker. What people call the pacemaker is the SA node. It's depolarizing intrinsically between, 80 to 100 times a minute. So to jump over to the PowerPoint, here's a graph from the book, and what they're showing us is you can see the depolarization of the SA node, how just following that, how the atria contract, and then I can see a depolarization of the AV node. Following that, the ventricles contract. And so basically, what you can see here is a difference in rate of depolarization, how the SA node is fastest, and how the SA node is the stimulus for the atria to contract, that when the SA node depolarizes, that depolarization will sweep through the atria, causing the atria to contract. And then once that depolarization hits the AV node, you see there's a bigger lag time because it's going to have to hit the AV node, go down the AV branches, and then go up the Purkinje fibers in order for the Purkinje fibers to make the ventricles contract. So let's take a look at a few more pictures. So what they're showing us here is when the SA node depolarizes, that depolarization is going to sweep into both atria. That's the signal for the atria to contract. That depolarization hits the AV node, and the depolarization is going to get shuttled past where the valves are located, because you might wonder, if we depolarize the atria, why not just, like rain or something, it just goes down over the ventricles, because the valves are located there. So the atria depolarize, and then that depolarization is shuttled into the interventricular septum, okay? And then notice this, how when the ventricles do contract, how that depolarization goes from the apex up, right? So when the ventricles contract, it's going to start from the bottom of the heart up, which is perfect, right? Because the semilunar valves are up where the AV valves are located. So here we can see the depolarization going down the AV bundle branches, hitting the bottom of the Purkinje fibers, and then up the Purkinje fibers, and the ventricles depolarize. That ventricular depolarization precedes ventricular contraction. So through these pictures, they're showing us a little tracing here, which is an ECG, an electrocardiogram. So because the depolarization, so I'm going to go back to the start of this, or actually let's look at this, let's look at the summary. Because the depolarization is an electrical event, what we can do, and I'm sure you've seen this or maybe even had this done, you can put electrodes on the surface of the skin and just measure the electrical changes that are originating in the heart. Because the heart is such a big organ, these electrical changes can be measured from the surface of the skin in a non-invasive way. That's what an electrocardiogram or an ECG is. And so if there's no electrical change, all we would see on an ECG is a flat line. I hope that makes sense. So this x-axis is just time going by. When there's a voltage change, that will create a wave, right? And the amplitude of that wave, the size of the wave, tells you about the size of the electrical event. We're not going to go into any type of interpretation of the EKG, so I'm not mentioning it for that. It's just, to basically understand what this is. So horizontally, you're looking at time, and then vertically, on the y-axis, you're looking at the magnitude of the electrical event. So when the SA node starts to depolarize, do you see how we'd be able to pick that up on an ECG? So I can see there's a little electrical change. And then when the atria are depolarized, you can see a little wave function occurred. That's known as the P wave. Okay, so the P wave is characteristic, and it tells us that the atria depolarized. When the depolarization goes down the interventricular septum, we can see a change, right? A little change. And when the ventricles depolarize, that's a much bigger chambers and a bigger event. We can see this big spike occur. That's called the QRS complex. And the QRS complex is a recording of ventricular depolarization. And then you can see how there's another wave, and what this is ventricular repolarization. So the ventricles go back to normal. That's called the T wave. And so to take a look at another picture, same story, right? when the atria depolarize, we can pick that up on the ECG as the P wave. And then what will happen is the message is going to have to get shuttled down the interventricular septum. So do you see how there's a little flat line there? So this is time going by in between when the atria depolarized, and we know that the ventricles are going to depolarize next. So now that the ventricles are starting to depolarize, we see the QRS complex. And then following QRS complex, we know that the ventricles are going to contract. But you see how this is time going by. And then what will happen is the ventricles are going to repolarize. They're just going to go back to the way they were. And we can see that on the ECG, and that's the T wave. So you might wonder, going back to this picture, that when the atria depolarize, I don't see a wave of atrial repolarization. And the atria do repolarize, and you could see that on an ECG, but that would be abnormal, because what should happen is that the ventricles depolarize fast enough that it drowns out the atrial repolarization. So atrial repolarization is also occurring with the ventricular depolarization. We just can't see it on an ECG. Now, if there was some type of heart block, like the message wasn't getting through, then we would be able to see atria depolarize, atria repolarize, ventricles depolarize, repolarize, and again, we would then know that something is wrong. Okay, so that would be abnormal. So going back to the notes, an ECG, all it is a graphic recording of electrical changes occurring in the heart, sensed by electrodes that are placed on the surface of the skin. So when there's a voltage change, we can see that as a wave function, and then the x-axis is just time. We should know the characteristic waves of the ECG. So the P wave is atrial depolarization, QRS complex is ventricular depolarization, and the T wave is ventricular repolarization. So to finish up, let me just talk a little bit about extrinsic innervation of the heart, meaning, the nervous system connecting to the heart. And I'm going to pull up a picture from our book. So here they're showing us that the heart does have nerves that connect from the autonomic nervous system. So the vagus nerve, you might remember, cranial nerve 10, innervates. You can see the SA node and the AV node. That's a parasympathetic nerve. And so that is going to work to decrease the heart rate. The sympathetic nervous system also innervates the heart. And so you can see the sympathetic cardiac nerves coming in green, also innervating the SA node, AV node, and other areas of the ventricles. The sympathetic fight or flight will increase the heart rate. So like we were saying before, the effect of the nervous system innervating the heart is to fine-tune the heart rate. And so if we severed all nerve connection to the heart, we said that the heart has the innate ability to beat. It does. The SA node is going to depolarize between 80 and 100 times a minute, and that should drive the heart at 80 to 100 times a minute. The thing is, our heart doesn't always beat at that rate. As a matter of fact, if you think of like your resting heart rate, is your resting heart rate between 80 and 100? I bet it's lower. And the reason that it's lower is because the parasympathetic nervous system is having an overall baseline effect on the heart. And so that's what the nervous system does. It influences the rate of the heart based on our activity. It can slow it down when we're at rest. It could speed it up when we're in a crisis. Okay, and so it's not just on autopilot. Our nervous system will help to fine-tune the heart rate. And so in the notes, I wrote that down. So nervous innervation will influence that intrinsic rate. So it's not really going to beat at that intrinsic rate. It could, but the sympathetic nervous system works to speed it up, and the parasympathetic innervation, which is typically, playing a role at rest, decreases our heart rate below that intrinsic 80 to 100 beats per minute. Thanks for watching.